Hello, I'm Gourab Ghosh Roy. I'm a third-year student doing my joint PhD at University of Birmingham and University of Melbourne. And today I'll be presenting our work, Bowtie Architecture of Gene Regulatory Networks in Species of Varying Complexity. So differences between species can exist at different levels. And our objective here is to understand differences between species in terms of some universally present dynamical gene regulatory system properties like robustness. And for that, we want to analyze the architecture of the gene regulatory network which supports these properties. One network architecture known to be associated with dynamical system properties is the Bowtie architecture, and it is observed in many different types of networks. It is formally defined by a largest strongly connected component, a core layer, between in and out layers. And a strongly connected component is a subnetwork in which every node is reachable from every other node. Regarding prior work, the existence of Bowtie architectures with a LSC core has been observed in eukaryotes like yeast and Arabidopsis. In our work, we investigate the existence of this architecture in GRNs of species of widely varying complexity from prokaryotes to unicellular and multicellular eukaryotes. And our objective is to analyze the differences in this GRN architectural feature with the aim of understanding the differences between species in terms of the dynamical gene regulatory system properties. So for our analysis, we have used GRNs available from public databases, and these are general GRNs, not specific to any context like experimental condition or cell type. And we have used the interactions between transcription factor genes and target genes, and we have used all interactions with any type or reliability ranking of evidence. And since we are looking for global architectures, we have used GRNs covering more than 50% of the species genes. There can exist many different definitions of biological complexity. So we have used this species complexity defined on the basis of the number of cell types, which we have obtained from literature. And based on this definition, we have arranged the species for which the GRNs are available in order of complexity as this, from E. coli to human. And here you can also see the sources from which the GRNs are extracted. So the Bowtie architecture with its 7 layers, core, in, out, the tendrils, tubes, and others, are defined like this, and with the core defined as the largest strongly connected component, the rest of the layers can be obtained through this decomposition algorithm. So we have applied this decomposition algorithm to the GRNs and obtained these results. Firstly, we see that there exists a core between in and out layers, and also this largest strongly connected core is much larger than the second largest strongly connected component present in the network. So depicting that in terms of distribution of nodes and distribution of regulator nodes in the different layers for different species, we firstly observe that a Bowtie architecture exists, and also there is a trend of a general increase in the relative core size with biological complexity. To look at how robust our results are, we have performed sensitivity analysis where we do Bowtie decomposition after random addition of 10% of edges to the original GRNs. And we also do this after random deletion of 10% edges. And in both cases, by plotting the average distribution of nodes and regulator nodes, we observe that the two aspects, that is the existence of the Bowtie architecture with the LSC core, and also the general increase in Bowtie core size with species complexity, both of them exist in both cases. Next, we compare our results with similar random networks. By similar, I mean random networks of similar node size and node degree. And here we plot the number of nodes of the original GRNs and also the number of nodes in the core of similar random networks in the box plots. And we observe that for E. coli and yeast, the original core size is significantly smaller than what is expected in random networks. And for more complex species, it is significantly higher. This division will be useful in our later observations or in our later hypothesis. So to summarize our observations, we observe the existence of the Bowtie architecture and also the increasing trend in Bowtie core size with complexity. And we also observe that the Bowtie architectures in these GRNs are characteristic features which cannot be explained just by chance. So now with our observations, we want to understand their biological implications. For this, we have looked at some dynamical system properties of interest. Robustness is the property of the system to withstand the effects of perturbations and maintain its functioning. And at the same time, you have fragility, where the system is being fragile to some unexpected perturbations. System flexibility denotes the property to support many possible outcomes. System criticality is the property by which it tunes to a point or a region of marginal stability. Controllability of a nonlinear dynamical system is the property to have a control path from an undesired equilibrium state to a desired equilibrium state in the phase space under the effect of finite perturbations. And evolvability is the property of the organism to generate heritable phenotypic variation. So using previously proposed associations of these properties with a bow tie core size, we hypothesize from our observations that a larger observed core in more complex species gives them increased robustness to perturbations not concentrated in the core, but at the same time, increased system fragility to specific perturbations concentrated in the core. It gives the system increased flexibility and moves it closer to criticality. But this comes at the cost of lower controllability. And in the long term, this gives the system increased evolvability. And we also speculate differences between groups of organisms, where in one side we have prokaryotic bacteria and unicellular eukaryotes, for which higher controllability is beneficial. And for more, and on the other side, you have more complex multicellular eukaryotes, for which increased flexibility and behavior closer to criticality, and in the long term, increased evolvability. All of those are important requirements. And all these requirements are supported by their respective GRN architectures. So in future, we want to test this hypothesis using dynamical models or maybe real systems. And we also want to investigate details of how the network bow tie architecture governs the network dynamics. These are some of the resources. Our paper has been recently published in the Journal of the Royal Society Interface, and also the data and code is available here. Thank you for listening. If you have any questions, please reach out to me. Thank you so much.